welcome to the National Cancer Research Institute in Birmingham, UK. You're from Bologna.
Yeah.
Tell us what you're doing there that's so interesting for the people in England.
We are very active in the field of diagnostic imaging, and in particular PET, using innovative, let's say, non-FDG tracer. That's to say, FDG is most widely and diffused tracer, which enable to diagnose cancer, essentially, when is overexpressing glycolysis. But in many cases, the tumor doesn't, just like prostate cancer, which is extremely common and very important. So our goal is to diagnose as soon as possible cancer, and in particular the non-FDG avid, like prostate. That's to say, when a patient has a relapse, a lab relapse, an increase of PSA, trying to be able to visualize where the recurrence occur in order to treat as soon as possible the patient.
So what isotopes are you using? Gallium?
Gallium is one of the many isotopes that we're using. For prostate, essentially, is choline that could be labeled with both carbon-11 and fluorine-18. But gallium definitely is the most rapidly growing isotopes of interest in that field.
So with choline you can do spectroscopy as well, magnetic resonance spectroscopy?
Yeah, the basis come from MR spectroscopy. Yeah. The idea was, let's say, stolen from the radiologist cousin or friend, right. But we use that labeled with isotopes in order to enable the imaging with PET. That's to say that you can whole body scanning the patient and exactly identify where the relapses occurred. So essentially we use carbon-11, but honestly, fluorinated labeling is much more interesting because of distribution. That means a widely available technique.
Sure. But you know, fluorinating an antibody or another molecule can change the molecule, can't it? And that's one of the criticisms.
Absolutely. It's not the case for choline, for sure, because it's small enough to be not essentially changed in its structure, so it reveals the membrane turnover, which is particularly rapid in prostate cancer and also in other cancers, just like, choline is also used in a central nervous system tumor, but we can also label, let's say, acetate with carbon-11 on F18, or the small peptides, the somatostatin receptor sensitive peptides, and they could be labeled, as you mentioned before, with gallium-68, which is rapidly...
And you use that in neuroendocrine tumours?
Absolutely.
Mm-hmm. Does it work?
Perfectly well. We are doing so far, I'm talking about routine application already available in clinics. I'm not talking about, let's say, cutting edge of research. We are doing on a weekly basis 10 to 12 patients with gallium DOTA peptides in order to diagnose the relapse and most of all to demonstrate the presence of receptors, which enables you for further treatment using the cold analogs or even the labeled with beta emitters like yttrium-90 or lutetium-177.
You've envisaged the time when, say, with choline, PET and NMR or MRI will converge?
I think it's more, to be very honest, a matter of competition among the two techniques, because essentially they will provide the same information. The key advantage to us is the fact that you can scan the whole body, while spectroscopy, PET can
scans the whole body.
Exactly. MR can only scan a small volume, so you can detect quite well the local relapse, but you cannot detect bone metastasis as well as lymph node relapse, which is quite important.
Thank you for coming and speaking to us.
My
Appreciate
pleasure.
it very much.